Austin, Georgia. The title of our talk is Zoledronate Inhibits Cell Membrane Repair. So please give Caroline a round of applause.
Good morning. My name is Caroline Lewis, and I'm a second-year medical student from the Medical College of Georgia. And I'm very excited to be here at the American College of Physicians Internal Medicine Conference to present my research project titled Zoledronate Inhibits Cell Membrane Repair. Now, before I describe my project, I wanted to give everyone a brief introduction to zoledronate. So zoledronate is an intravenously administered drug of the bisphosphonate class. And this class of drugs inhibits osteoclast function to promote bone mass increase. And so it inhibits resorption of bone, increasing the mass of bone. So this class of drugs is used to treat a variety of disorders, such as osteoporosis, Paget's disease, hypercalcemia of malignancy, and others. And the dosing varies with the pathology being treated. And while these drugs work quite well in treating these disorders, they're associated with a very poorly understood, rare, but very serious side effect known as osteonecrosis of the jaw. It's abbreviated BRONJ, also known as bisphosphonate-related osteonecrosis of the jaw. And this is what BRONJ looks like. Obviously, a very serious side effect, and although rare, we really don't understand why this side effect occurs. And we decided to study zoledronate and its role in cell membrane repair because this side effect is actually initiated when patients being treated with the drug long-term undergo a dental procedure in which there's some sort of trauma to the jaw, such as a tooth extraction or any other dental procedure where there's a trauma to the jaw. And so since the side effect occurs upon a trauma event, we thought that a repair mechanism may be inhibited. And so we also saw other studies had shown that other bisphosphonate drugs, such as atidronate and zoledronate, inhibit protein machinery in the cell membrane repair pathway. And so we hypothesized that zoledronate inhibits cell membrane repair. And the way we study cell membrane repair is that we blast cells with a laser. And you can see the injury on the periphery of the cell. And this is actually healthy repair. So the way we measure the degree of repair is the fluorescence. And so when repair is successful, so the cell membrane repair heals, or the cell membrane heals, and very little fluorescence can enter the cell. And so right here you see very healthy repair, and although this is a very non-physiologic injury, the cells can actually withstand a blast by the laser. And so very little fluorescence, this is healthy repair. Now when the cells are treated with zoledronate, there's a very severe uptake of the dye. And these cells, because they cannot seal that membrane, they cannot maintain internal homeostasis, and they die. So this clearly shows that zoledronate inhibits cell membrane repair in this cell type, which is a mouse myeloblast. And so side by side, these images speak for themselves. Zoledronate inhibits cell membrane repair. And we can demonstrate this graphically by measuring fluorescence on the y-axis and time on the x-axis. And the lowest green line right there is healthy repair, and obviously very little fluorescence, so the cell membrane is healing, and these cells survive. The lines in between are all the cells treated with zoledronate at various doses. And we also saw the same exact response in another cell type, and this is actually mouse kidney epithelial cells. So in conclusion, zoledronate inhibits cell membrane repair in two distinct cell types. And we think this may be important in this side effect because cell membrane repair is a very important mechanism in wound healing. Because as cells glide past one another to heal a wound, there's a lot of friction involved, and cell membranes tear very frequently. And if they can't repair these cell membranes, the cells die, and wound healing cannot occur. And since the BRONJ side effect is inhibited by, or is caused by trauma, we think that there's a failure of wound healing occurring. And another important thing to note about these images that I showed is that zoledronate is not causing any kind of cytotoxicity. If we could go back and look at those cells when they were initially treated before the injury, they're actually very healthy. They still have all their cell processes attached to the bottom of the plate. And so the drug's not actually killing the cell. It's preventing the cell from mending the fence on the periphery. And so it really is suggestive that it's actually inhibiting a wound repair mechanism that is cell membrane repair. And so obviously this is a very complex side effect, and but we think that this may be one part of the picture of this very poorly understood side effect. And since we found that zoledronate inhibits cell membrane repair in two distinct cell types, we thought that future research should focus on other cell types and to see what the effect is of the drug on those cell types, but also studying possible prophylactic treatments. And recently, vitamin E, a very readily available, non-toxic, well-tolerated material or substance, has been found to promote cell membrane repair. So we propose that future research should possibly focus on treating, perhaps, cells being treated with vitamin E to prevent this, or to promote cell membrane repair. So we think that that is a possible prophylactic treatment, but in conclusion, zoledronate markedly inhibits cell membrane repair in two distinct cell types. Thank you very much.
Thank you, Caroline, for the wonderful presentation. Very interesting research. Any questions from the audience? We'll go over there. And then we'll all go back.
Different doses of zoledronate depending on pathology. Have you noticed a correlation with osteoporosis being more often found in patients who are treated for more benign versus more malignant?
Well, I'm assuming you're talking about the side effect bronch, like have the incidence. Well, the incidence of bronch is not surprisingly, it's highest in patients who are receiving the highest doses. So hypercalcemia, that is the pathology that's treated with the most, it's usually treated with intravenous form, which is zoledronate. And it's, I can't remember the exact dose, but it's treated most frequently. So osteoporosis, I think if you're being treated with an intravenous drug, you may receive an injection once a year. But for hypercalcemia, you can receive injections as often as once a week. So that's where the highest incidence of the side effect is occurring, is in hypercalcemia patients. But the actual incidence of the side effect is actually unknown. So there's been a lot of different studies, but we actually don't know the true incidence of the side effect.
Really interesting talk. So I was wondering at the in vivo concentration that you achieve when people are dosed with Zoledronate, and whether your cell culture model, is that like a 1 micromolar, is that an approximation of what you see in vivo?
Well, we try to do some sort of approximation, but it's quite difficult because the drug itself, it really does go to the bone, and we really couldn't find in the literature very accurate numerical values for what molarity we use or whatnot, but we try to make it as close to the dose as possible.
Caroline, it seems like based on your studies in the kidney epithelial cells and myoblasts that this particular drug inhibits cell repair. How does this relate to, say, for skin traumas?
Right. Well, to be honest, the reason we use these cells is because that's what it was available in the lab, as research often goes. But it is important. I think these cell types are relevant because, particularly myocytes, muscle cells, whenever we do any kind of exercise, muscles glide past each other, and the friction between the muscle cells creates cell membrane tears. So any cell in the body where there's a lot of friction, such as skin for wound healing or muscles, there has to be a lot of repair occurring or the cells die. So future research, I really think it's important to try, oral epithelial cells, or osteoblasts, whatnot. But I think these cells are relevant because they also have to, in order to survive, they have to have so many cell membrane repairs occurring.
Thank you for the wonderful presentation.
Congratulations.